Welcome to the Performance Around the Clock podcast. I am Professor Satchin Panda, a scientist in the field of circadian rhythm. In this podcast, I will talk to experts in many different fields about science, health, and human performance. You'll also find out why circadian rhythms is my favorite topic. I hope you enjoy the podcast and thank you for listening. Welcome to another episode of Performance Around the Clock. I'm super excited to have Professor Geoff Wahl as my guest today. Those of you who may not know, Professor Wahl has a long history of very foundational research in cancer. He worked at the Salk Institute for over 45 years?
46 years.
46 years. Working on cancers, various types of cancers, and many of his discoveries have gone on to translation. That means they have been used to manage, prevent, and cure cancer. Dr. Wahl was also the president of the American Association of Cancer Research. And as president, he also was very active to increase awareness among the public for the terrible toll of cancer and how to raise funding from grassroots level for research on cancer. Welcome, Geoff.
Sachin, it's a pleasure to be back at the Salk and to speak with you.
Yeah. When I talk to people about cancer, the first thing almost everybody understands what is cancer, because our lifetime risk is almost 40%. And
yeah, I did an analysis this morning in preparation for speaking to you, and the classic numbers were lifetime risk is 50% for men and 33% for women. Of course, that changes in different countries as well. But now, overall, it's about 50%. I think that's because we're diagnosing it more frequently.
So that means, for example, for a couple, there is a chance that one of the couple will get diagnosed with cancer in their lifetime.
Yes. But I think another way of looking at it is to really ask the question, what fraction of people will have a cancer experience in their lifetime? Mm-hmm. That means they may not get it, but they will almost certainly know someone who has it. Because having a cancer experience in your lifetime can make a profound difference, especially in people who may not have adequate insurance coverage. It can lead to bankruptcy. It can lead to enormous stress. So these are a group of diseases, not one disease. It isn't cancer, it's cancers that impact everyone in very significant ways, whether you get the diagnosis or not.
So when people say cancer, and you say this is a group of disease,
right?
Can you tell us what are cancers and why they're so deadly?
So not all cancers are deadly, but what cancer is we start from a single cell. The egg and the sperm meet, and then the cells divide like crazy. And then there are controls that prevent the cells from dividing further. That's why we don't have livers the size of the earth, right? They're a certain stereotype size. Cancer is a disease within cells that changes its ability to sense those controls about when it should stop dividing. So instead of the cells stopping dividing, they continue to divide. And then we can divide them broadly into two classes, those that are benign, a benign tumor, or a malignant cancer. A benign tumor is not typically dangerous. I say not typically because they can be, depending upon where they develop. But usually the cells kind of more or less look like normal. They stay in one place. They don't move to different sites. Their genetic material is, changed, but not in dramatic ways. And a surgeon can go in, and if the tumor is in an accessible site, can remove it, and the chances of the tumor recurring are very, very, very low. Okay? Cancer is a different story. Cancer generally refers to a cell that grows out of control, grows faster than those in a benign tumor. The most important thing is that those cells that are cancerous and malignant, that means that they have a good chance of moving outside of where they started, for example, the breast, to distant organs, for example, the bone, the lung, the brain. And in those distant sites, they can continue to grow, and that's what causes death. Generally, it isn't the cancer in the organ itself, like a cancer in a mammary in a breast. If it just stayed there, it may look ugly, but it wouldn't kill a person. But once it moves to a different site, that's a different story. So it's that metastatic spread, the movement from one place to another, that's most lethal.
But what about cancers inside the brain?
So
Those tumors.
So cancers inside the brain, again, can be of two different types. They can be benign or they can be malignant. The benign ones generally stay in one spot. They can grow to be quite large. They can cause dysfunction, neurologic dysfunctions in a person, but once excised, often those functions can return. A malignant cancer in the brain typically doesn't spread to other organs. It's kind of constrained within the brain because the brain has certain barriers around it that prevent cells from moving outside of it. But by moving to different parts in the brain, they can really wreak havoc and lead to lethality. In fact, glioblastoma, which is the most dangerous cancer of the brain, has a very, very short 5-year survival rate, a very small 5-year survival rate. So it's a very dangerous cancer.
So I always ask this, what made you to come work on cancer?
What made me work on cancer? So I think, let me go into a slightly different question and I'll get to the cancer part of it. What made me get into science? And what made me get into science was that I myself had a very serious illness when I was a kid. And this illness was a meningitis caused by a virus, so an inflammation of the surface of the brain caused by a virus. So there was no treatment for it, and there was a 50% mortality rate. So in fact, I was in the hospital for quite some time, and I was in the bed next to a kid with exactly the same illness. And it turned out that he died and I lived.
Oh, sorry.
And so I always thought from that point on, I survived for a reason. And I attribute my survival to the doctor that I had, who took very aggressive measures, which I really didn't appreciate at the time, but they saved my life. So I decided that I would do something related to helping people. And I wasn't sure what that would be, but I was out of school for quite some time, and my science teacher was the first one to show great confidence in me. He said, "Geoff, I'm going to give you an A because I know from your performance thus far, you would have gotten an A anyway. I'm not going to make you do any makeup work or anything like that. You're going to get an A. In fact, I'm also going to recommend you for the Science Olympiad," which I did terribly. Because they asked about a bunch of stuff that involved memory and fossils and things like that, which I had no interest in. But I had a great interest in biology. And then through school, I remember vividly in high school, there was an issue of Life magazine that was the first one where I could see the central dogma of biology. DNA goes to RNA, goes to protein. And the images that were shown were so vivid, I said, I can understand disease because I can understand it from the genetic basis all the way up to the functional element, the protein. That's what I want to do. So I ended up going to UCLA, where I work with a chemist. And I did organic chemistry with him.
It's undergrad.
Undergrad. And I actually was showing off in the lab one day to the woman who would eventually become my wife, not my current wife, but a first one. And I threw some sodium metal into the sink to show her how a metal could create a flame, which I thought was really pretty amazing. But what I didn't realize is that somebody had put a flammable solution into the sink before I threw the metal in, and so the whole sink started on fire, which I put out with sand, which was the right thing to do. Apparently, the professor must have learned about this, because he soon introduced me to a friend of his who had just come to UCLA, who was a biochemist. Maybe he thought that biology was a safer place for me to be. And again, I had fabulous mentors. They believed in me. They gave me intellectual freedom. They gave me very challenging projects to work on. But I could see that I could formulate an idea, a hypothesis. And they gave me the tools to learn how to test that hypothesis. And so from that point of working with this biochemist, David Sigmund, who had just come from Harvard, he said, "Geoff, you're going to Harvard." And I said, "Great, I've never been out of California." And so I got into Harvard. We don't have to go into the story there. But the man that I worked for ultimately developed the technique for genetically engineering mice and received the Nobel Prize for that, Mario Capecchi. Again, the reason I got into Mario's lab was that I had been in his class and he asked us to write proposals, like a grant proposal. And I wrote this proposal, and he was so intrigued by it that he asked me to come into his lab to work on that idea. That was empowering. And of course, I couldn't make it work. It failed. But on the other hand, I learned how to design experiments, and if they didn't work, I learned perseverance. And then I went to Stanford, and that's when the molecular biology revolution was really taking off. And so we were one of the first ones to do molecular cloning, and I learned then the power of that technique for solving problems. It could solve the problem that I had originally designed to do in Mario Capecchi's lab that I didn't have the tools for. And that led to the Salk Institute, where I've been for 46 years. But why did I get into cancer? That was your question. Yeah. The reason was because after I had survived this illness, a good friend of mine's uncle, who I had been very close to, had an adult onset leukemia, which killed him very rapidly. And it was a horrible death. And I remember seeing that. And then after I had gotten here, my father was hospitalized. They thought it was a heart problem. He ultimately died of cardiac failure. But as part of the workup, they did a chest x-ray on him. It turned out he had a huge lung cancer. Oh. And if he didn't die of this cardiac failure, he definitely would have died of that lung cancer. He was a lifelong smoker. And then fast forward a few years, my mother thought that she had flu. I was at a conference in Greece. My wife, who's an oncologist, told me, "You better come back right away." I came back. My brother, who's a doctor, and I went up to see her, and he palpated her abdomen. It turned out that she had colon cancer. So here are two cancers, one lung, one colon, one that could have been prevented by a person not smoking or having gone to the doctor more frequently, getting chest x-rays and seeing something earlier. The second clearly being preventable by colonoscopy. And so I had always been interested in how cells become cancerous, how a normal cell acquires the ability to change so rapidly through both the genetic changes that it undergoes and then other processes. And so the stimulus provided by seeing this uncle die a horrible death in his early adulthood, and then seeing my dad and my mom get two cancers, solidified my direction into doing something in cancer that could help people in the future.
Wow. Actually, I didn't know about this background. And as we started, you mentioned that almost everybody will have
A cancer experience.
in
Yeah.
their lifetime.
Yep.
And now I see how it pans out in everybody's life.
Exactly.
Yes.
And you know, I had initially thought about being a doctor,
Yeah.
but my experience in the hospital gave me white coat syndrome because I had so many, you know, invasive procedures done on me. So instead, I decided after my first marriage didn't work out, I met my wife in the lab, who was an MD PhD student and with whom I've been married for 47 years.
But
she's a breast oncologist. And so she teaches me what I really need to know to be a better scientist. She teaches me about the physiology and the complications of cancers in humans.
Yeah, so this also segues to my other question. That is, you said that you wanted, at one point, you thought about becoming a doctor. And as a doctor, you could have been an oncologist treating cancer patients. Then you decided to come and do research. So for general audience, can you discuss what is this basic research on cancer versus, when sometimes I go and once in a while I would have a cancer project and I would tell people would ask, what are you working on? Cancer. And then the first thing that comes to their mind is, which cancer are you going to cure?
Yeah, right.
Which drug?
Right.
So what's the difference between this basic research on cancer versus becoming a doctor to treat cancer or finding a drug to treat cancer?
I'm glad that you asked that. And I'm glad that you use the word basic because it's a word that I hate. We should never call what we do basic research because basic implies it's easy. You know, you should know about it already. It's basic, right? Instead, I call it foundational.
Yes.
Imagine a house. The foundation is the first part that's built. If that part isn't strong, durable, that will hold up the rest of the structure for the life of the individual and even beyond, then what worth is it? So foundational science is what we do. It's creating that level of understanding that enables us to take that information forward so that we can understand processes, biological processes, diseases, cancers at their root, so that we can develop more wise, not just sensitive, but wise ways of early detection, more effective ways of preventing cancers because we understand the biology, we understand what the risk factors are, or if necessary, treatment. So one of the reasons, so there were two reasons why I decided to go into foundational research versus being a doctor. One, I've already said, I'm squeamish, I can't stand blood. The more important reason is that I thought, you know, if I can discover something foundational, that will stand the test of time and could impact people in perpetuity. It could impact so many people. And even if I never discover a cure for a particular cancer, that wouldn't matter because the information that I generate will be useful for subsequent generations. And an important subcomponent of that is mentoring. We must always be able to train and inspire the students of today to become the scientists and mentors of tomorrow. I felt if I could be a clinician, I could have more immediate impacts. I could affect some number of people, but the number would be discrete depending upon my lifespan and the number of patients I could see. And also, quite honestly, I felt I wasn't wise enough to understand the biology well enough to never make a mistake. And I felt if I ever made a mistake on a patient, I couldn't live with myself. And that's why I have such reverence for my wife and what she's done.
Yeah. No, I guess when you're talking about this foundational, one thing that came to my mind, I think, for example, cancer for a very long time was thought to be caused by viruses or something else. And then
Can be, by the way. as well as bacteria.
Yeah. But I think one of the key idea that I think almost all cancers are due to genetic mutations in DNA.
Mm-hmm.
And that foundational discovery
Yeah.
has led to the discovery of which genes are mutated.
Yeah.
Or, for example, now the most rapidly rising detection method to
Yeah.
check whether you have cancer or not is based on just looking for those mutations.
Right.
And it's also, like you said, finding new drugs that will treat specific mutations in specific genes. So I guess that's one example where.
So that's the idea of personalized medicine, right?
Yeah. But it all goes back to the foundational discovery.
Yes, absolutely.
That cancers are due to genetic mutation.
So, so again, we need to, I think broader. So, how a normal cell gains that ability to stop responding to the signals that tell it to stop growing and to stay where it is. Yes, the initial changes in the DNA change genes that alter the ability of the cell to perceive its environment or its conditions. And those genes are frequently mutated. But remember, there's the important thing about cancer is its ability to spread to distant sites, to metastasize. And the initial assumption was that in order for a single cell to become cancerous, it goes through multiple stages in which multiple mutations are accumulated, 6, 7 mutations over a person's lifespan. And if a person smokes, getting back to my dad, smoke has carcinogens in it. Those are chemicals that interact with DNA and change it. So the probability of a person who smokes getting lung cancer that's dangerous is very high, significantly increased. So, but that doesn't address the question of, well, how does the cell move from its site of origin to a distant site? And actually, this is a huge unmet need right now. We don't know. It used to be thought that this same kind of multi-step process of mutation accumulation is what's involved. But you know what? As we've gained the ability to sequence DNA, right? That's where all the secrets are. That's the DNA is the book of life. Each gene is a word. The word it gives the instructions of what a cell is supposed to do. People thought, okay, well, we need to change 5, 6, 7 words for the cell to become a cancer to grow uncontrollably. Then we need to change another 5, 6, 7 words in order for the cell to go from its normal site through blood vessels and colonize distant sites. Let's look for those mutations. Haven't been found. Have not been found. And furthermore, our ability to sequence the DNA very sensitively now, which is one of the great benefits that's come from cancer research, these methods of fast, cheap sequencing. We now know that the cells that just start out and are in the breast, and the cells that start to move, so they've gained that ability to move from their normal site, they have the same genetic mutations. They're not different. And some epidemiologic studies say when that first cancer starts, a metastatic cancer is already there, can be, like in pancreas cancer. So that raises the interesting possibility that it's the microenvironment acting upon those mutations that have already occurred that instruct the cell, you can move to a new site. But you know what? Historically, 90% of the money that's gone into cancer research has been spent studying those primary tumors that we can excise surgically, not the metastatic ones that cause people to die. So we've got to change the equation, studying the things that really cause mortality. So yes, there are mutations. There are common ones like P53 or RAS or many other ones. that are part of this instruction mechanism for cell growth or response to conditions that impinge on the cell. But then there's this whole black box of the environment in which the cell is growing.
But if we go back to this idea that, yes, there are mutations that decide the cell, that makes the cells divide a little bit faster and make a tumor. In our lifetime, there might be a lot of mutations happening. There might be a lot of tiny tumors starting. And then maybe our immune system goes and checks for them and then kills them. And
Yes.
similarly, is the immune system also checking for all the migrant cancer cells and somehow neutralizing them? And metastasis is a failure of the immune system or surveillance.
So that's a very important point. What about the immune system? And what can we do about that? Is there some way that we can harness it? So, there's this famous statement that it is not the brightest of species or the strongest of species that are able to survive, but rather the most adaptive.
Yeah.
That was attributed to Darwin, but Darwin actually didn't say it. It was Leon Megginson who said it, and he was a business professor. And the reason that he said it is that he was trying to understand how businesses under challenging conditions could survive. And so he started to read evolutionary theory. And so he came upon Darwin and he said, this is what Darwin meant. Why am I bringing this up? Because you've got two adaptive systems in conflict with one another. You've got the cancer. For a cell to grow uncontrollably, it's changing its environment in which it's surviving. And so it's got to be developing mechanisms that enable it to adapt to those changing environments. Then you've got the second powerful adaptive system, and that's the immune system. So the immune system is looking for things that are different from self, different from what it's used to. And the cancer has mutant proteins, and those mutant proteins can activate the immune system. So, in fact, probably the reason that not everyone dies of cancer is because the immune system is pretty active in keeping it under control. In fact, studies have been done, for example, on men that are a bit older than I am, in their 80s, who have died not because of cancer, but of some other reason. And then autopsies have been done on them, and they've looked to ask the question, did they have any evidence of prostate cancer, for example? And it turns out that more than 80% of men who are 80 years old have prostate cancer. It didn't kill them. They were surviving with it. And
Well, let's repeat that again. What did you say? 80?
More than 80% of men who are 80 years old or over will have evidence of prostate cancer. They won't die from it. They'll die with it. They'll die from something else. Okay?
Wow, that's a
Stroke
very
or something like
profound
that.
number.
Oh, yeah, absolutely. Same is true of breast cancer. So you can, in fact, one of the problems with breast cancer is that we're getting better and better and better at diagnosing it at earlier stages. So there's something called ductal carcinoma in situ. Carcinoma sounds like cancer, cancros from the cancer. Is it a cancer or not? It turns out most women with ductal carcinoma in situ would be just fine if they weren't treated. The problem is we don't know which will be fine. And that's an important topic of current research. How can we predict which of these women who have this tumor growing in their breast are more likely to develop metastatic disease? Okay? So the immune system and cancer in tension with one another. The cancer is trying to develop mechanisms that quiet down the immune system. And so that's where this new field, well, it's actually probably been in development for the past 2 decades, of targeted immune therapy is coming in. So it turns out the cancers can develop mechanisms that attenuate the immune system to prevent the immune system from interacting with them. There are checkpoints, and these cancers are very clever, and the immune system says, oh, I think I see a cancer, but the cancer cells, at least some of them say, oh no, nothing here to see, and then nothing happens. And so that's when that cancer becomes very dangerous. There are a number of mechanisms for that. One of the ones that actually our final papers are on is very exciting. And that is that some of the earliest mutations that enable cancers to grow in the first place enable the adult cell to reprogram, to change its state, to revert, to reprise, to reacquire the characteristics that are found early during development. Why is that important? Because in order for this nascent organism, the fetus, to grow, the fetus is actually very different from the mother, right? Because it's got the father's genes. So actually, it's kind of like a tumor in that sense. So in order for that fetus to grow, you have to develop, the fetus has to develop immune evasive mechanisms. Some of those involve the way that the fetus is actually connected to the maternal blood supply. But other ones involve the changes that go on, the genes that are expressed that attenuate the immune system. And guess what? Cancers reprise those same mechanisms to fool the immune system. So they reprogram into this early embryonic-like state. Immune evasiveness. And
So,
yeah,
go ahead.
no, you.
No, I was thinking, we covered.
That came from, by the way, foundational studies.
Yes.
Foundational studies.
So now that has been used
development.
in many aspects of cancer research. So now we discussed quite a few points, or you alluded to quite a few points, how mutations start cancer, and then how start tumors. Not all tumors are cancerous.
Right.
Some tumors move, and then some cancers are lethal.
Right.
And over the last 40 plus years, you have been working on breast cancer.
Right.
And let's put all of this back into the context of breast cancer. And let's discuss how 40 or 50 years ago, breast cancer was almost a death sentence.
Some, yeah.
And what are the major discoveries or milestones that happened in those 40 or 50 years? Because people now can connect how foundational research
Thank you.
led to, if we use breast cancer as an example, how it has helped to reduce mortality from breast cancer, and how now at least few million people are cancer survivors, breast cancer survivors, and they're living a near normal life.
Yeah. So great example, great question. So let's go back early on. Let's go back to the
70s?
1900s.
Right.
In the 1900s, if a woman came in with a lump, the surgeons would initially just take out that tumor from the breast. And then there was a surgeon, I think his name was Halsted. What he found was that even if that tumor was taken out, a lot of women would die. That gets to this notion that they already had metastatic disease, okay? But he didn't know that. What he thought is, well, we need to use larger and larger and larger margins, okay? So that's where the idea of radical mastectomy came in. But in a large study that was done, I think in the 60s or 70s, maybe more recently, I'm not an oncologist, so I don't know those particular clinical data. But what I remember is that a study was done to look at the recurrence rate on people who had lumpectomies versus radical mastectomies, and they were basically the same. So that said, you don't really need a radical mastectomy. That was a huge difference because they were largely disfiguring, very challenging surgeries to get better from. And so it led to the idea that, well, we can take out the primary tumor, but there's obviously another reason that these women are dying. The disease must have already disseminated to different sites. So now we're going to do surgery in combination with chemotherapy. And the chemotherapies were designed to kill rapidly dividing cells. That's what cancer is, right? Fine. And that led to some improvement. But it also made the assumption that all breast cancers are the same. Well, with advances in molecular medicine and in actually the first studies just used antibodies to see whether breast cancers expressed or didn't express estrogen receptors and progesterone receptors, the two growth factor sensing receptors that enable the breast cells to grow. And then the doctors could classify the cancers into two different groups. There were those that are hormone receptor positive and hormone receptor negative. And now what they could do is the hormone receptor negative ones you could treat just with chemotherapy. The hormone receptor positive ones you could also treat with compounds that prevented the signaling from those growth promoting receptors, right? Or you could give women drugs that prevented the synthesis of estrogen and progesterone. Now, even those drugs can be very challenging for some women to take. And then there was another group of women who didn't express the estrogen or progesterone receptors, but they expressed something else that was causing them to grow. And again, foundational biology showed that that was another kind of growth factor receptor called HER2, the human EGF-related receptor 2. So it's a growth factor receptor that's part of a family.
So
yeah.
Let's take a pause and explain what is these three hormones and why they're important, or how the cells are using these hormones, hijacking.
Okay, good. So during the normal growth of the breast, at puberty, the breast really undergoes extensive growth. So there's a ductal system that's designed to make milk. And before puberty, it's kind of rudimentary. But after puberty, when a woman starts to have her period and is now capable of reproducing, the breast tissue, the ducts grow dramatically. They grow only because the girl, now become a woman, is producing these hormones, estrogen and progesterone, that interact with molecules on the cell that tell the cell to divide. And so then you get a rapid expansion of the ducts, and you have enough material that's required to produce the milk for the baby, okay? That's one class. The cancer uses those very same mechanisms, right? Cancers don't develop new things, they use old things in new and perturbed ways. Then there's another kind of growth factor called the epidermal growth factor. There are a series of these growth factors. Their name tells you what they do. They control growth. And normally they're present at very low levels on a cell. But in some of these cancers, they're produced at much, much higher levels because the gene that encodes the instructions for making them is produced in much higher numbers. Now, it turns out that was a subject that I studied in the first 10 years at Salk, figuring out how that process happens. And it turns out there's a guy named Dennis Slayman who figured out that it was what's the protein is called HER2, that's the receptor that receives these growth factor signals, is amplified or produced at elevated levels in this subset of breast cancers that at the time were death sentences. So that's about 20% of all human breast cancers have elevated levels of this HER2 protein due to amplification, increased copy number of the HER2 gene. What Dennis Slayman recognized is he could make an antibody that attached to that receptor and stop it from signaling. So that was one of the early, not the first, but one of the early examples of personalized cancer therapy. And there's a whole wonderful story about that, how that drug was developed and how it almost died and all that we don't have time to go into. But needless to say, what that took was foundational biology. Dennis Slayman was an MD. He worked with foundational biologists who had cloned the genes and then could produce enough of this protein to make the antibodies that they could then use to treat human patients. And that also took an advocate, Fran Visco, who actually had what's called HER2 positive breast cancer that was metastatic, and she desperately needed this drug. She went to the Department of Defense, and she's a tenacious lawyer, and she got them to give the money that they did not spend on weapons to put into the research that was needed to develop the drug that now is being used to treat many women and now has made HER2 positive breast cancer, instead of a death sentence, one of the most eminently treatable ones. Okay, so now we've gone from
when did
Halstead,
that happen? What is the timeline of this HER2 therapy? 80s?
The 80s and 90s and early 90s. And now there are a whole bunch of different products that are even more effective than that drug Herceptin that was initially designed. So now we've gone from Halstead, who did these radical mastectomies, to lumpectomy with chemotherapy. And now we've subdivided the breast cancers into multiple groups, into what's called triple negative. So they don't have the estrogen receptor, the progesterone receptor, or the HER2 gene amplified. Into the estrogen receptor, progesterone receptor positive ones, those can be treated with not only chemotherapy, but also with anti-estrogen or androgen therapy, anti-progesterone therapy. And then we've got the HER2 positive that can be treated with anti-HER2 agents. But it's more complicated than that. Because even this group that's called triple negative, the one that's negative for everything, it's not really negative for everything. It's positive for some things. And now we're starting to learn what it's positive for. For example, there's a subset that have repurposed the androgen receptor, which is usually expressed in men, is now expressed in women. And they have enough androgen that they can actually cause the cells to divide. But it gets more complicated than that, because each of these tumors, the tumors within an individual woman, the cells within that individual tumor are all different from one another. They're heterogeneous. And this is part of the way that they fool the immune system. And so this, what's called intratumoral heterogeneity. So there's interpersonal heterogeneity, the different classes of tumor, of different classes of breast cancer. And then there's intratumoral heterogeneity that refers to the differences in characteristics of individual cells within the tumor. It's that intratumoral heterogeneity that defeats personalized medicine. Because how can you develop a specific personalized drug if that person's tumor, the cells are different from one another? And that's where you need combination therapies. You need therapies that will hit the majority of cells in the tumor that you know about. And then you need adaptive mechanisms such as activating the immune system to take care of the rest. And I often, when people say, oh, I'm cured of my cancer, I try to gently say to them, you have no detectable disease. To imply to them that you must be vigilant, because even a single cancer cell, given enough time, can grow back to a dangerous tumor. So vigilance is important.
So let's go back to this intratumoral heterogeneity.
Yeah.
So you gave an example how HER2 positive breast cancer patients can get Herceptin,
Right.
And in many cases it can cure them. So kind of quote unquote cure.
Actually, let me correct you there.
Yeah.
Because what does HER2 mean, right? How does a doctor classify a tumor as HER2 positive? Turns out if only, I think it's 5% of the cells turn out to be HER2 positive by molecular tests, they say, okay, we're going to treat that person with Herceptin.
Is that the threshold, like 5%?
Yeah.
So the pathologist looks at the slide and then
Yeah. And they go, well, maybe some of the other cells are below the level of detection of our methods. But it's also likely true, in fact, is true, that a lot of the other cells, because of the way the tumor evolves, don't have HER2 at all. And in fact, those are the ones that become selected for growth once you treat a person with a drug like Herceptin. So what do? You can't treat with Herceptin alone. You have to treat with Herceptin plus chemotherapy, or Herceptin plus immune therapy, or Herceptin plus chemotherapy plus immune therapy. And then it becomes a game of whack-a-mole. This is where early diagnosis becomes critically important, because the larger the tumor, the more it's spread, the greater the chance that there's going to be variation among all those different cells that generate what we call that person's metastatic cancer. And within larger populations, that gives you a greater chance that one of them is going to be resistant to whatever you throw at it. So early detection is best.
so it's almost if I'm thinking of an analogy, it's like a small gang in a neighborhood, and there is a gang leader, and that can be the dominant HER2 positive, for example, in this case, and the drug is given to take out the HER2 positive cells, but then the other gang members can rise up the ladder and then can kind of continue to create trouble. And if the group is small, then it's easy to control them. But if this,
It's kind of a Martin Scorsese way of looking at it, but yeah, okay.
well, sometimes you got to bring.
I think, yeah, I think whack-a-mole is an effective visualization tool, because you hit one and then another one pops up.
Yeah.
That's where this idea of coming up with mechanisms that are as adaptive as the cancer is critical.
So right now in 2025, for example, let's dial back. What is the lifetime risk of a woman getting diagnosed with breast cancer?
So a woman who has no predisposing genes, it's somewhere between 33 and 50%.
Without predisposing gene.
Yeah.
So these
lifetime risk.
So these are BRCA
negative,
negative, P53, all this negative.
Right. And this is related to environment.
Yeah.
There are risk factors for development of breast cancers, and there's also racial factors that are involved in that. And those racial factors can be both due to access to effective medical care, but can also be due to environmental factors. For example, there are many people where incidence is determined by, pollutants in their environment. And that's where, you have to look at cancer clusters and things like that.
Yeah. So now, since there is this 33 to 50% risk, how much of that is...
And I should mention, it used to be thought, classically it was thought, well, cancer is a disease of old age, right? You live longer, you have a greater chance of accumulating mutations, this and that. Turns out that the most alarming thing I've seen recently is a dramatic increase in the frequency of cancers in younger people, and especially colon cancer in women and men, and breast cancer in women. And we do not understand the reasons for that at all, but it's likely to be environment-based.
Yeah. No, actually, this relates to what I work on, circadian rhythm, and in fact, there is now many studies, including one by IARC, classifying shift work or chronic circadian rhythm disruption, increasing the risk for certain cancer, not all cancers, and colon cancer and breast cancer is on their list.
Yeah.
So.
Yeah, and I'll bet that has to do with stress factors, at least part of it.
Yeah. So now coming back to foundational research helping, although we have 50% risk of being diagnosed with cancer, and you mentioned the word early detection. So what is your opinion on early detection? What should women do about early detection? And in your opinion, means you have been on AACR, so you must have seen all kinds of numbers. Roughly, how many lives or how many cancers are detected and treated most effectively because of early detection? Because this is one huge part, because as you said, when the tumor is small, if you can detect and treat, that can be most effective, more effective than when the tumor is large.
I think that, this is really a question to ask my wife because she's really has been involved in this for her entire career. But clearly, breast self-exam is, and teaching women to do that effectively is extremely important. Many women actually will be the first ones to find evidence of it. Classic mammography is giving way to much newer and more sensitive methods of detection. But again, it's not just early, but it's wise. So we're detecting more cancers. Does that mean we're doing better on the survival curves? And for some cancers, yes, and for many cancers, no. So we need to be able to be wise in how we analyze these cancers and determine who needs treatment and who's going to do fine with just watchful waiting. Because with every treatment, there's a risk of some side effect and mortality. So you don't want to overtreat, right? And you don't want to undertreat. So that's where wise diagnosis comes in. And that's where the foundational research is so critical. We need to understand the biology better. We need to understand the factors to look at. It's possible that with AI and with pathologists, not AI alone and not pathologists alone, but the combination of the two of them, that features will be detectable that we didn't realize were important before, because now you have the world of data at your fingertips that can be analyzed instantaneously to look for patterns that you didn't see before. So I'm hopeful that that will be able to inform us of who needs to be treated and who doesn't. There's a very famous small animal pathologist named Bob Cardiff. And he can actually look at a tumor, a mouse mammary tumor, equivalent of a breast cancer, and he can tell you just by the looks of that tumor what gene created it.
Wow.
Yeah, because they have very stereotyped patterns. Yeah. I think that that kind of information is going to be emerging with the combination of AI, molecular analysis, small-scale DNA DNA sequencing, and pathologic analysis, probably within the next 5 to 10 years.
Yeah, no, that's exciting. The whole AI has been transforming research in multiple fields, and
we have to be careful of it, because it hoovers all the information from the internet, and a lot of that information is bad. So we have to be able to distinguish good, reliable information from bad garbage information.
Yeah. Now, I guess one point that you discussed, we touched upon briefly, was how cancer cells and the very primordial cell that gives rise to us, like from a fertilized egg to the whole baby human, the similarity between these two. Like they all start from one cell, they divide and one becomes a tumor, and one becomes an organ or something else. And similarly, we talked about at puberty, how these couple of hormones trigger a cascade of changes that start maybe from some stem cells lurking in the...
Oh.
So how are the stem cells different between cancer, or the cells that give rise to cancer versus the stem cells that have the promise of giving rise to an organ?
Oh, Satchin, you're wading into murky waters here. Well, so you're talking about, I think you're trying to get at this idea of cancer stem cells.
Yeah. And also the reason why this is important also, because the stem cell therapy holds a lot of promise, but at the same time, there is this risk that some of these stem cells can become cancerous?
Okay. So these are two very different things. So cancer stem cells, the idea was that the question was, are all cells in a tumor equally capable of generating a new tumor? Or is it that there's a very small subset of cells within that tumor that are the originators of that tumor, the stem cell of the tumor? And the way the experiment was done was to, in mice, take a tumor and dissociate it into individual cells, and then separate the cells on the basis of some of their characteristics, usually by markers that were expressed on the surface of the cell. And then ask, were cells that were high in marker 1 and 2 equally capable of forming a tumor as cells that were high in markers 3 and 4, or was there a difference between them? And it turned out there could be enormous differences between them.
Mm-hmm.
But as time is, and so people call those cancer stem cells, and then some people said, oh, that these cells were like the stem cells of the organ from which the cancer arose. So we've actually done a lot of work on this problem, and it turns out that the system used for analysis is fraught. Because what you're doing is you're taking a cell from a tumor, and you can't do this with a human, because if you do it with a human and you do it in a mouse, you have to use an immune incompetent animal. So immediately you get rid of the immune system, so that has no bearing. Most of the time, you're putting these cells into a site in the animal that has no relevance. And you're also taking cells that have gotten tons of genetic changes in them already, which has no bearing on what the cell looked like in the very start. Okay? And finally, as we've become more and more able to analyze which cells are actually the stem cells of an organ, turns out that the cells that are able to form tumors in animals by this transplantation assay have no bearing genetically, molecularly, to the cells that form the stem cells of the organ. There are a few exceptions to that, okay? And the exceptions are in the colon and in the brain that I know. Those are the best characterized ones. But in other cases, no. And why is the reason for that? Well, it turns out the last thing you want to do is carry stem cells throughout your lifetime, because these are cells that are geared to divide rapidly and then differentiate quickly in order to form the various structures within an organ. The only organs in which those cells are designed to persist are in the colon, because you're sloughing the cells all the time, you're being exposed to toxins, etc. The skin, where the same thing happens, maybe some in the lung, but generally, most of the cells in the organs of our body are not dividing at all. When do they divide? They divide when there's a wound and you need to repair the damage. And it turns out that in the skin, in order to repair the wound, the cells actually reprogram to this embryonic-like state so that they can rapidly repair that wound and close it, okay? It turns out in the breast, we've recently found that something similar happens, okay? This is a normal cell. If you transplant this cell, so now you've taken it out of its normal environment, it rapidly reacquires the fetal-like state where we know the only stem cells for the breast are localized. That's something we showed a long time ago. And now we have all the genetics, so we could actually say, okay, well, part of this process of regeneration is the same thing. So what keeps normal cells from doing that? It's the microenvironment that constrains them. It's the cell-cell context that they normally experience as an organ grows. Once you disrupt that, which is actually something that happens during cancer development, cancer has been called the ever-healing wound that never heals. And so that's why These cells can reprise these embryonic-like programs. They can start to evoke these immune evasive mechanisms, and that's what enables them to grow into a tumor to begin with.
I see. So then from the other side, stem cell therapy.
So stem cell
therapy.
You're not going to use stem cell therapy for cancer. You're
No,
going
no,
to
no.
use stem cell therapy.
Stem cell therapy for other reasons.
So it used to. So in the early days, people would use a series of genes to reprogram any adult cell into a more primitive state, that then you could expose the cells in that primitive state to different conditions, and you could get them to differentiate, to turn into a brain or a skin, now a mammary gland or other thing, a kidney, a pancreas. But it turns out that the genes that were being used are genes that cause cancer, right? So you really don't want to do that. Now people have been able to replace those genes with either chemicals or other methods that would not be carcinogenic, not induce cancer. So you can do that. And in fact, it's been tried now in the blood system for people with sickle cell trait, where you can take the cells, the stem cells of the hematopoietic system, a person's normal stem cells, they have a mutation in the gene that causes the sickle cell trait. Now you can use gene editing tools to correct that mutation. And now you can put those cells back in. And because they were stem cells to begin with, they'll repopulate the entire the blood system with normal cells. Now there's a problem with this, because even the gene editing mechanism is not 100% accurate. And so you worry about other changes being created that you didn't intend, but that have happened. And because these cells are going to stay with the person for the length of their life, the possibility of those turning into a cancer. And we need to have more people studied. I think the sickle cell disease is so awful and there's no effective treatments for it, that the people are willing to take the chance on doing that. There are other places where people have congenital blindness that can develop progressively through life. The eye is an immune sanctuary. So you can put cells into the eye to replace defective ones to regain sight, but those cells are going to stay there. They're not going to move elsewhere. So that would be a safe place to do it. And there may be other cases where you can do genetic modification, maybe even on a stem cell, and put in mechanisms that constrain the growth of that cell or prevent it from growing beyond a safe point. So to put in kill switches. So I think that these are going to be things that are going to be used in the future, but I've got to get to the issue of funding.
Yeah.
Proposals are now being made to decrease funding at the NIH for foundational science of the type that we've been talking about by 30%. This has never been seen before, never. And if the idea is to just hand off all of the foundational research to pharma or biotech and hope that they're going to do it, that's not going to happen, because the research I've been talking about is long-term and risky. And so that's the purpose of the government, is to fund the kind of research that creates the solid foundations for the future, where drug companies can take the findings or the drugs that are developed in the labs and turn them into the drugs that are safe to use in people.
Yeah, so coming back to funding, you have been at Salk for 46 years. And during that time, Salk actually has a NCI-funded
Cancer center.
Foundational cancer center.
Mm-hmm.
And you have much better idea about the history of cancer research at Salk, foundational cancer research. And can you give a couple of examples where those foundational research have actually moved on to trigger pharma to put money into finding cure that are actually in clinic?
Well, before I retired, I was the Daniel and Martina Lewis Professor. And why was I that? Why those two names? Well, it turns out that Dan Lewis has chronic myelogenous leukemia. Chronic myelogenous leukemia used to be a death sentence. It would go into this thing called blast crisis and then rapidly kill a person. It's no longer a death sentence. Dan Lewis has been living with it for decades. Why is that? Well, he's been given a drug called Gleevec. Well, what does Gleevec do? Gleevec attacks the protein, inhibits the protein, the enzyme that is created by this genetic mutation, let me put it simply, that drives the growth of chronic myelogenous leukemia. How is this aberrant gene found? Well, it was found by the foundational research done in several places, among which was at the Salk Institute by Tony Hunter. So Tony Hunter didn't develop Gleevec, but he found out about a class of genes that turns out to be present in viruses that cause cancer. And those genes are also mutated in many people with many kinds of cancer. And one of these genes that's mutated is the one that's found in chronic myelogenous leukemia. So here you have a full cycle event. Work that was done by Tony Hunter, Dan Lewis knew about, and it was because of that work that he attributed this life-saving medicine to. Ron Evans has done foundational work on a class of receptors for hormones that control virtually most of the processes in the body, and this has led to the development of many, many drugs in many different companies. Satchin, I don't know if the work that you've done on circadian rhythm has led to the development, if not necessarily drugs, but of strategies to treat people with sleep disorders, or in your case of even time-restricted feeding, of helping people in a way that prevents or limits, reduces obesity, and puts people into healthier phases of their life. And similarly, many studies have been done in neurobiology here that have people with neurologic diseases. So work that was done by Greg Lemke, who kind of started his career as a foundational scientist, discovered many of the same class of enzymes that Tony Hunter developed, has now led to discoveries of a group of enzymes that are frequently mutated in cancer, as well as in other diseases, that has now led to drugs that are undergoing clinical trials. So, and this is a small institute. So if you look at bang for your buck, I mean, every dollar that's put into an NCI designated cancer center generates at least $3 in return revenue.
But I guess it's more in this case.
It's got to be much more.
No, whatever you said. Like, for example, if we go back and say how much of NCI, National Cancer Institute funding, came to Tony Hunter's research up to the discovery of, tyrosine kinases and the mechanisms, it must be less than few tens of millions of dollars.
Yes.
And then if you look at how that led to pharma putting money into that to come up with a drug, and now that drug, this pharma has made a lot of money, but that's okay.
Well, and that's generated wealth within the community.
Yeah, and so many people benefited just working for the drug, marketing the drug, and then other stuff. But I guess the most important point is, from that foundational research, if we now connect the dots, there have been at least few million people.
Oh.
At least a few million people.
Oh, no, tens of millions of people.
Tens of millions of people. So that's why I think we should not...
And don't count it just in the US, count it worldwide.
Yeah, so we should not think of $1 converts to $3.
No, right.
No, $1 actually equivalent to saving one life.
Or many lives. Let me give you another example. And this isn't a drug. This is a concept. So Renato Del Becco was given the Nobel Prize for figuring out the connections between viruses and cancer, which also was part of the same Nobel Prize that was given to people studying viruses that cause cancer, retroviruses that cause, now lentiviruses that cause AIDS. Renato Del Becco said, to understand cancer, we have to be able to sequence every single gene. So he wrote a paper in Science. That single paper in Science, opinion piece, 1 page long, led to the development of the Human Genome Project. The development of the Human Genome Project led to the sequencing of the first humans. That led to the technologies, and that sequencing of the first human genome, all of the Book of Life, was, I forgot how many millions of dollars, maybe $100 million.
No, 3 billion. The first genome.
3 billion, sorry, $3 billion.
Yeah.
So call it $3 billion. And now we can sequence that same person for $1,000, $2,000.
Less than that, even now.
Less than that.
$300.
And faster. That took years. Now it takes days. One of the things we did in my lab was I met a breast cancer survivor, okay, who turned out to be an advocate for breast cancer research. I met her at a meeting. Turns out that her sister had 4 recurrences of breast cancer. She was in the hospital at the same time with her first diagnosis at the same time as her sister was having her 3rd recurrence. When this woman was in my lab, she had a recurrence. My wife saved her life. I said to her, geez, we should sequence your DNA. You may have a gene that's predisposing your family to the development of breast cancer. And through advocacy work here and through a bike ride that raised money for breast cancer, we were the first ones to sequence a genome in San Diego, her genome. Turned out she had no genes that we could identify that would have been the usual suspects for calling breast cancer. That doesn't mean that she doesn't have one. It just means that we couldn't find it. Moral of the story is 30 years ago, we never could have dreamed of doing that. Now it's something routine. So how valuable is that? I mean, that whole technology, you know, think about NCIS, you know, think about, you know, solving cold cases, think about early detection of cancer. All those things were made possible because Renato Del Becco had the vision, the wisdom of saying, we need to develop ways of rapidly sequencing whole human genomes. Remarkable.
And Renato was a professor here,
yeah,
for the record. And yeah. So when you were AACR president, you also started this Stand Up to Cancer drive.
Well, don't say I started it.
You are very humble.
I was asked by Marge Foti on my first meeting of the foundation board, what do you think, you know, so what ideas do you have for how the AACR could make a significant impact in the future? And I said, and that was a time of another funding crisis. And I said, well, if Jerry Lewis can have a telethon for muscular dystrophy, and that affects very few people, and cancer affects virtually everyone, as we've discussed, certainly there's justification for having a grassroots effort to raise money for cancer research. And in fact, I said, just think about it. Everybody is touched by cancer. There are 300 million people in this country. If every one of those people on average gave $10 a year, that would raise $3 billion a year, which at that time was 60% of the money being given to the NCI. I said, that would make an enormous difference. It needs to be grassroots. Now, Sherry Lansing, who was the head of former head of Paramount Pictures, said, so you're talking about a telethon for cancer. That's the only way you're going to be able to get it to be broadly transmitted. I said, yes, exactly. She said, I'm going to go to Hollywood to see if they're going to be interested. And in fact, people in Hollywood were thinking the same thing because there was one woman who was the head of one of the studios who actually was diagnosed with breast cancer, a very malignant form. And so she desperately needed this research to be done. And that's actually how the Salk Institute got started in its pancreatic cancer research efforts, was because one of the first grants given from Stand Up to Cancer was to do pancreatic cancer because it's such a lethal cancer. And one of the places that was funded was the Salk Institute. So, you know, I'm happy that that was generated, but we desperately need that now. We need an independent source of funding beyond the government because we can see now how fragile governmental funding is.
No, I guess it's not only it's the philanthropic support should not be a substitute for government funding, but it can actually trigger new ideas, just like you said. We got into pancreatic cancer because of this foundation, Stand Up to Cancer funding. And in my lab, there are multiple examples where even small donations, just like you said, $500 right now can enable any lab here to get a genome sequenced. Just imagine, $500. It was a government project, $3 billion 25 years ago, now $500. And you said $10 from every person, 300 million people. That's what I always say, that if people can just pay for 2 cappuccino.
Yeah, I often talk about that. So let's talk about how much we actually spend on cancer research.
Yeah.
So right now, it's about $7 billion for the National Cancer Institute. There are 300 million people. If you do the math, that's, $25 per person, right? Per year. Yeah. $25 per person per year for diseases that are going to affect everybody. So how many Starbucks mocha cappuccino, large, could you get? Maybe 3 or 4, depending upon where you're buying it, right? So 3 or 4, is that really all that we're spending for diseases that will affect everybody? And think about how much the treatment of each of those people costs. It's obviously thousands of fold more than that, okay? And it's taking people in the prime of their life. Now, as I said, cancers are being diagnosed, lethal cancers are being diagnosed earlier and earlier. So clearly, we need to understand why, we need to mitigate the causes, we need to be able to come up with wiser diagnoses and more effective treatments for these very common diseases. And the other thing is, if you just use your own AI chatbot and ask the following question: how many lives have been saved because of research funded by the NIH versus how many lives would have been lost had that research never been done? And look and do it over the period 1990 to the present. And you'll be stunned.
Should we do it now?
Go ahead and do it.
Okay.
Use Perplexity.
Oh, I don't have a...
Whatever.
Okay.
You can use Claude, or you can use ChatGPT. I actually have that graph, but...
Okay, so you can actually...
Well, I have it. I actually gave it to Michelle Chamberlain here. Oh, you're fast at typing, Satchin. Don't make errors.
Let's see.
The curves go like this.
So, according to American Cancer Society, 2023 Annual Cancer Statistics Report, the US cancer death rate dropped by 33% from its peak in 1991. So that means every 3 people who were diagnosed or who would have died in 1991,
one of them now lives.
One of them now lives.
Imagine if that's your child.
Yeah.
Imagine if that's your parent. Imagine if it was my mother.
Yeah.
She would have lived to see the bar mitzvah of my son and my daughter, bat mitzvah of my daughter. She didn't live for either of those.
So it roughly translates to 150,000 lives saved every single year.
Right. So multiply that by and again, every one of those lives is irreplaceable. Imagine if one of those lives that had been saved was Steve Jobs.
Yeah.
Maybe we'd have iPhone 27 now. Maybe anyway, one can only imagine what the impact would be. And again, it's a small investment for a lifetime of impact.
Yeah. Any parting comment, any end comment? What gives you hope for cancer research now?
What gives me hope is the unbridled enthusiasm and perseverance of the young people who we're mentoring. They deserve the same opportunity as we had to pursue their dreams.
Thank you. Thank you, Geoff. There was an exciting episode, and I'm sure we can again come back and discuss about cancer because this is such a complex and big topic. And as you said, it will touch everybody's life in our lifetime. So knowing very fundamental things about cancer also gives us the power of knowledge that when we face this danger in our life, then we can have a better understanding how to detect, what conversation we should have with our doctor, and how to even live as a cancer survivor. Thank you.
Thank you, Satchin.